Hello, my name is Ruiz. The project I am working on is promiscuity of the enzyme Tautomerase in the biosynthesis of natural products in strain Caulobacter endophytes of cycad Dioon edule. Natural products are structurally diverse chemical compounds with the multiple bioactivities. Their biosynthesis requires that a minimum of genes to be expressed, also known as a biosynthetic gene cluster or BGC. Studying microbial natural products under an evolutionary and phylogenomic approach will clarify whether in the family of genes occurred on the evolutionary history some events of duplication, horizontal transfer, or mutation, and define if these events engage in the curve of certain enzymes to be recruited within the specialized metabolism. The diversity of specialized metabolism is also the result of substrate selection during enzyme activity. Many enzymes can be promiscuous, catalyzing more than one on the biochemical reaction of their substrate promiscuity. However, enzymes can go through some mutation and become specialized to recognize only one particular substrate over the different metabolic pathways leading to the biosynthesis of new natural products. For this reason, it's important to explore different taxonomic groups to discover and understand the biosynthesis of new natural products. In my biological model, we are exploring the genomes of two Caulobacter strains that were isolated within the corollary root of cycad Dioon edule. The genomes of these endophytic strains were annotated using anti-smash, where we identified a region with 80% similarity to individual BGC. We performed an alignment of the canonical individual BGC reported in the Tautomerase chromofuscus strain with our individual-like BGC, and two differences were notable. The first, the INV gene in our Caulobacter is smaller, and the second, the existence of a small gene named CoB between INC and INA gene. CoB encodes an enzyme of the 77 amino acid related to the Tautomerase family. Using bioinformatic tools Corazon and BSK, we have observed that the Tautomerase domain and a similar individual-like BGC is shared between our Caulobacter strain and some actinomycetes. However, it should be noted that each of the actinomycetes synthesizes different natural products, despite their non-ribosomal peptides recognize the same residues glutamine at initial substrate. In bacteria, many of gene clusters that encode non-ribosomal peptides also encode accessory protein of the MDTH-like protein superfamily, which are small proteins of about 70 amino acids. These proteins alter the profile of the substrate in the annihilation domain in non-ribosomal peptides. Therefore, we hypothesize that this tautomerase could act as a protein to MDTH-like, an enzyme that participates in the substrate selection process in individual-like BGC. To begin to understand the function of the tautomerase, we predicted the tridimensional structure of CoB monomer holding the structural domain alpha beta alpha using open source ColabFold and AlphaFold packages. We predicted 12 possible contact sites with the catalyzed region for substrate recognition. This area is painted blue using the tool P2Rank. The ligand will mainly be of the site of the smaller alpha helix. After analysis in P2Rank, we tested several amino acids at possible substrate with CBDOC2. The best score was selected based on the best Viña score, which is calculated from the free energy generated between the contact of the ligand and the receptor. These values indicate that the lowest score offer a better prediction. Tryptophan residue had the highest score, while tyrosine was the amino acid with the highest contact with residues but bound into the catalytic site. We also evaluated indigwinin and noted that the molecule directly competed with substrate to inhibit the catalytic site of tautomerase. Based on the lab observation, we propose that this tautomerase prefers molecules with aromatic rings as substrate, and indigwinin is a potential regulator in the biosynthetic step. Our next goal is to corroborate that point by expressing and synthesizing the